Imagine embarking on a high-altitude trek, eyes fixed on the summit. The air thins, pushing your limits, and suddenly, well, you hit an invisible wall. Debilitating exhaustion washes over you, your muscles scream, your mind clouds, utterly unable to focus. It's a total bioenergetic collapse, A profound and unexpected loss of power, leaving you stranded. Now contrast that vivid, punishing scenario with the seasoned mountaineer. This individual, facing the exact same thin air and punishing ascent, powers through. Their steps are steady, breathing controlled, focus on wavering. What's their secret? It's not just sheer willpower. It's robust cellular machinery, humming efficiently beneath the surface, adapting to that extreme stress. This striking contrast, this hidden biological edge, sets the perfect stage for today's deep dive.
Indeed. That prepared mountaineer embodies a body where the internal powerhouses are functioning optimally, even under extreme duress. And that's precisely what we're going to unravel today. Our core topic is how the relentless march of aging profoundly affects these cellular powerhouses, known universally as mitochondria. We'll also explore their intricate, sophisticated quality control system, a vital process called mitophagy. Our mission is clear. We aim to understand how reactive oxygen species, or ROS, can surprisingly both signal cellular adaptation and, paradoxically, inflict significant damage. And we're diving headfirst into one of the most hotly debated topics in longevity science: the age-related decline of NAD, a molecule absolutely central to life itself.
Okay, and a critical distinction before we truly begin. While we've certainly touched on cellular cleanup before in a previous deep dive, our focus today is laser-sharp on mitophagy. That's mitochondria-specific autophagy, right? The cell's highly targeted process for getting rid of only the faulty or surplus mitochondria. We're not discussing general proteostasis or broader autophagy today, except in those moments where mitophagy is directly and integrally involved. This allows us to really zero in on the unique challenges and solutions for our cellular energy centers.
Exactly. This deep dive is all about the power plants of your cells. Our narrative will trace from that initial altitude sickness crash, our vivid metaphor for cellular energy crisis and dysfunction, to the empowering concept that targeted quality control for mitochondria, combined with a proper and consistent fuel supply, could potentially restore and then sustain your vital energy capacity throughout life.
The aim is really to move beyond merely enduring, towards genuinely thriving, much like that well-prepared mountaineer we pictured. It's going to be a comprehensive journey. First, we'll thoroughly explore how the process of aging impacts mitochondrial function, leading directly to reduced energy output and an increase in cellular stress. Then we'll take a deep dive into mitophagy, the cell's sophisticated cleanup crew, specifically tasked with removing damaged mitochondria, examining its crucial pathways in detail. Next, we'll uncover how dysfunctional mitochondria can inadvertently trigger widespread inflammation throughout the body, a process that researchers are calling inflammaging. After that, we'll pivot to one of the most powerful and accessible interventions. How regular exercise acts as a mitochondrial elixir, promoting both the creation of new healthy mitochondria and the efficient cleanup of old damaged ones.
Following that, we'll confront the NAD question head-on, why NAD levels decline so significantly with age and its critical implications for overall cellular health, including, of course, mitochondrial function. This leads naturally into the emerging and often controversial field of NAD boosters and the mixed evidence surrounding their efficacy in humans. From there, we'll shift our focus to an even more upstream strategy, targeting CD38, a major enzyme primarily responsible for the consumption and depletion of NAD. We'll then briefly touch on some other recently emerging longevity compounds like taurine and alpha-ketoglutarate, dissecting the immense hype versus the actual evidence-based reality.
And a crucial nuanced concept that truly reshapes our understanding will be mitohormesis. We'll explain why a little bit of stress, even something like reactive oxygen species, can actually be profoundly beneficial, and why, counterintuitively, too many antioxidants can sometimes backfire. Finally, we'll explore the practical biomarkers for measuring mitochondrial health, giving you objective tools to understand what's truly happening at a cellular level. We'll also address the critical reality of conflicting data and important safety considerations in this rapidly evolving field, culminating in actionable takeaways for you, our listener, to apply to your own life.
Right. Our goal throughout this deep dive is to provide intellectual rigor, guiding you through complex biological mechanisms with clarity and precision. We aim to equip you with an actionable, evidence-based understanding, allowing you to critically discern genuine scientific advancements from mere speculation in the fascinating realm of aging and cellular health.
Let's begin by talking about mitochondria themselves. We often describe them as the cellular engines, and it's a perfectly apt analogy for a reason. They are the primary architects of ATP, which is adenosine triphosphate. Think of ATP as the universal energy currency, the ready cash that powers virtually every single cell, every process, every thought, and every movement in our bodies. From the delicate firing of a single neuron to the powerful contraction of muscle fiber, ATP is what makes it all happen. It's fundamental.
And this is where the profound impact of aging truly comes into play. With advancing age, there is a demonstrably consistent trend of declining mitochondrial function across various tissues. This isn't just a minor dip in performance, It manifests as a significant reduction in that crucial ATP output, leading directly to lower cellular energy availability. Imagine an aging power plant that becomes less efficient at producing electricity and starts to draw more power just to keep its own lights on. Simultaneously, older mitochondria exhibit increased oxidative stress. They produce more reactive oxygen species, or ROS, than their younger counterparts. This creates a vicious cycle where the very act of generating energy also generates more damaging byproducts, perpetuating further damage within the cell.
That decline isn't an abstract scientific concept. It's something many of us have observed or experienced firsthand. This direct reduction in available cellular energy contributes, quite profoundly, to the pervasive fatigue and increased physical frailty that are commonly observed in elderly individuals. It's not just about feeling tired. It impacts daily function, limiting mobility, reducing the capacity for physical activity, and ultimately diminishing overall quality of life. Even simple tasks like climbing stairs or carrying groceries can become significantly harder, all rooted in this underlying cellular energy crisis.
Exactly. And at a molecular level, this decline is linked directly to the accumulation of various forms of damage within the mitochondria themselves. Specifically, we observe significant mutations and deletions in mitochondrial DNA, or mtDNA. This is a unique genetic material distinct from the DNA in the cell's nucleus, residing exclusively within the mitochondria. These mutations and deletions impair the integrity and proper function of the mitochondrial genome. Furthermore, proteins within the mitochondria can become oxidized, similar to how metal rusts, further compromising their precise structural roles and their ability to carry out enzymatic reactions. This accumulated damage directly impairs oxidative phosphorylation, or OXPHOS, which is the very complex process by which ATP is generated using oxygen. This impairment demonstrably reduces, for example, muscle endurance, making sustained activity increasingly difficult.
And researchers haven't just theorized this. They've consistently observed specific, measurable changes in older muscle tissue. There's a clear increased prevalence of those mtDNA mutations and deletions, strongly suggesting a genomic instability within these organelles as we age. Accompanying this, higher levels of ROS are detected, indicating elevated oxidative stress within the aging muscle cells. And crucially, a reduced oxidative capacity is consistently observed, directly linking these specific molecular changes to a decline in the muscle's ability to efficiently produce energy using oxygen.
Right. These findings, collectively and strongly, suggest a fundamental decline in mitochondrial quality control mechanisms as organisms age. It's as if the self-repair and maintenance systems become less vigilant and less effective over time. To counter this, the body employs two primary strategies to maintain a healthy and functional mitochondrial pool. The first is biogenesis, which is the process of making brand new mitochondria. Think of it as regularly building new power plants to replace older ones or increase capacity. The second, which is a key focus for us today, is mitophagy, the highly selective removal of damaged or surplus mitochondria. This is the body's targeted cleanup operation, ensuring that only the most efficient and healthy power plants remain in service.
Okay. But despite these built-in maintenance mechanisms, the evidence increasingly indicates that mitophagy activity itself can become dysregulated as we age. So it's a significant challenge. Not only is there more damage accumulating in mitochondria, but the cellular cleanup crew isn't as efficient or responsive as it once was.
Precisely. For instance, basal mitophagy, that's the routine background level of cleanup, particularly via the crucial PNK1 Parkin pathway, shows partial impairment in older cells. This impairment manifests as an accumulation of PNK1 and Parkin proteins. Normally, these proteins are involved in tagging damaged mitochondria for removal, but their buildup suggests a bottleneck in the process, a failure to clear them effectively. We also see reduced mitochondrial ubiquitination, which means the necessary molecular tags for degradation aren't being applied effectively. The direct consequence of this impaired tagging and accumulation is a decreased clearance of defective mitochondria. These dysfunctional mitochondria then remain within the cell, continuing to produce harmful ROS, consuming valuable resources, and actively contributing to more cellular damage, thereby accelerating the very phenotypes we associate with aging.
So, in essence,
Aging mitochondria are caught in this kind of spiraling decline. They produce less energy, generate more harmful reactive oxygen species. And critically, the body's sophisticated quality control systems, mitophagy for cleanup and biogenesis for renewal, struggle immensely to keep up. This creates a cascading effect of cellular decline, impacting overall health and function, and bringing us back to that initial feeling of hitting the wall during our high-altitude trek. That's a fundamental challenge of biological aging.
That's a perfect summary of the situation we face.
Okay.
Micro recap point 1, approx 5 minutes.
Okay, let's just pause for a quick recap. We've established that mitochondria are our cellular powerhouses, making ATP. But with age, they decline, producing less energy, more damaging ROS. This leads to things like fatigue and frailty.
Right. And this decline is linked to accumulated damage, like mutations in mitochondrial DNA and oxidized proteins. Critically, the body's cleanup system for mitochondria, called mitophagy, also seems to become less efficient with age, letting this damage build up. That's the core problem we're unpacking. Got it. So damaged power plants and a less effective cleanup crew. Let's dive deeper into that cleanup crew now, mitophagy. Well, mitophagy. We've established that mitophagy is essential for clearing out damaged mitochondria, especially as we age. But what exactly is mitophagy, and how does the cell pull off such a precise, targeted cleanup operation? How does it know which mitochondria to target for removal?
That's a fundamental question about cellular intelligence, isn't it? Mitophagy is a highly specialized form of autophagy, often translated as cellular self-eating. But unlike general autophagy, which might indiscriminately degrade various cellular components, mitophagy is remarkably selective. Its sole purpose is the targeted removal of faulty, surplus, or even just old and inefficient mitochondria. It's a precise, sophisticated mechanism honed over eons of evolution, designed to maintain a perfectly healthy and highly efficient mitochondrial population, ensuring peak cellular performance. The cell employs two main initiation pathways, both fascinating in their molecular choreography. The first, and arguably the most well-studied and crucial pathway for mitochondrial quality control, is the P-INK1 Parkin pathway. Imagine this as a sophisticated molecular tagging system. Under normal, healthy cellular conditions, a protein kinase called P-INK1 is rapidly imported into healthy mitochondria and quickly degraded once inside. It's like a constant quality check happening all the time. However, when mitochondria are damaged, perhaps by oxidative stress or metabolic overload, they often lose their membrane potential. Think of this membrane potential as the electrical charge across the mitochondrial inner membrane, a key indicator of its health and functionality. This loss of potential is a critical signal. It prevents PNK1's normal import and subsequent degradation. So instead of being removed, PNK1 stabilizes and begins to accumulate on the outer mitochondrial membrane, or OMM. It's like a warning light flashing on the exterior of a faulty engine. This accumulated TNK1 then acts as a beacon, recruiting another crucial protein, the E3 ubiquitin ligase called Parkin. Parkin normally resides in the cell's cytosol, the internal fluid. Once recruited to the damaged mitochondrion by PNK1, Parkin becomes activated. What Parkin then does is a process called ubiquitination. It attaches small ubiquitin proteins as tags onto various proteins located on the outer mitochondrial membrane. These ubiquitin tags are like molecular red flags or degradation signals. They specifically mark the distressed mitochondrion for removal. This ubiquitin signal then attracts specific autophagy adapters. Key examples include P62 and OPTN. Think of these adapters as molecular bridges. They connect the ubiquitinated mitochondrion to the machinery of autophagy. These adapters facilitate the formation of a specialized structure called an autophagosome. These are double-membraned vesicles that literally begin to engulf the tagged mitochondrion. Once fully engulfed, the autophagosome fuses with a lysosome, which is the cell's recycling center, filled with powerful digestive enzymes. The damaged mitochondrion is then thoroughly destroyed, and its components are broken down and recycled back into the cell for new synthesis.
Wow, that PINK1 Parkin mechanism sounds incredibly precise. It's like the cell's internal waste management system has a very specific set of instructions for which power plants to decommission. And it's not just theoretical, is it? We see consequences when this goes wrong.
Absolutely. It truly is a remarkable system. To underscore its importance, consider a stark real-world example. Mutations in these very genes, particularly PINK1 and Parkin, are notably linked to familial Parkinson's disease. In these genetic forms of Parkinson's, defective mitophagy leads to the harmful accumulation of dysfunctional mitochondria, especially within neurons. This mitochondrial dysfunction contributes significantly to the neurodegeneration characteristic of the disease, highlighting just how vital this pathway is for neuronal health and overall cellular integrity. But it's not the only way. Beyond this, cells also possess receptor-mediated pathways. These are alternative mechanisms where specific mitophagy receptors are located directly on the mitochondrial membrane. These receptors can directly trigger autophagy independent of the ubiquitination tagging process. Specific examples include proteins such as BNIP3, NIX, and F1NDC1. These receptors are typically induced under specific stress conditions such as hypoxia, a state of low oxygen. Their mechanism is quite elegant. They directly bind to LC3, a protein integral to autophagosome formation, thereby initiating the engulfment process without needing the parkinubiquitin steps. These pathways often operate in a tissue or context-specific manner. For instance, BNIP3 NIX-mediated mitophagy is vital in the maturation of red blood cells, ensuring these cells shed their mitochondria, and also plays a critical role under low oxygen conditions in other tissues.
So in youthful cells, there's this delicate dynamic balance between getting rid of the old and making the new. It's like a perfectly maintained factory with an efficient production line and a meticulous waste management system, ensuring a consistently healthy and highly efficient mitochondrial pool.
Precisely. That's the ideal state. However, as we age, the picture becomes more nuanced. There's increasing evidence suggesting that mitophagy efficiency can decline in some tissues, leading to a problematic buildup of those damaged, inefficient mitochondria. For instance, one study focusing on primary human fibroblasts found that their baseline mitophagy was indeed dependent on the PNK1 Parkin pathway. When this crucial mechanism was experimentally suppressed, those cells began to exhibit features characteristic of aging, underscoring just how important this cleanup process is for maintaining cellular youthfulness. But here's where the nuance and complexity, which are so characteristic of aging research, truly come into play. A more recent, highly comprehensive analysis utilized a sophisticated fluorescent mitophagy reporter in mice that allows researchers to literally see mitochondria as they're being degraded. What they found was surprising. Mitophagy is not universally downregulated with age across all tissues. In some organs, it was unchanged, or remarkably, in others, it was even increased. This indicates a far more complex tissue-specific response to aging, rather than a simple across-the-board decline. Interestingly, that same study also identified an increase in inflammation, specifically type I interferon responses, in aged tissues. This inflammation was linked directly to the presence of cytosolic mitochondrial DNA, which is mtDNA that is leaked out of the mitochondria into the main fluid of the cell. This is a crucial point that we'll elaborate on in our next section. But here's the therapeutic punchline. Crucially, this study demonstrated that pharmacologically boosting mitophagy using a compound called urolithin A in old mice resulted in tangible, observable benefits. It successfully reduced the inflammatory activation and, even more importantly, significantly improved the physical function of these older animals.
That's fascinating. So the implication here is profound. Enhancing the cell's mitochondrial cleanup mechanisms can indeed alleviate certain age-related declines. This really positions mitophagy as a highly promising therapeutic target to restore mitochondrial quality and, by extension, cellular vitality in the context of aging.
Absolutely. And it's vital to understand that mitophagy doesn't work in isolation. It functions hand in hand with mitochondrial biogenesis. As old or damaged mitochondria are efficiently degraded, new ones must be made to maintain the cell's energy supply and overall metabolic capacity. Key regulators of this biogenesis process include master transcription factors like PGC1, NRF1, NRF2, and TFAM. They orchestrate the creation of new mitochondrial components and their assembly. Beyond biogenesis and mitophagy, there's another critical quality control layer, the mitochondrial unfolded protein response, or UPRM2. Think of UPRMT as an internal distress signal system within the cell. If mitochondria experience stress, for instance, if their proteins aren't folding correctly or there's a mild degree of dysfunction, they send signals back to the nucleus. This nuclear signaling then orchestrates a specific response. It upregulates chaperones and proteases, which are proteins that help refold or degrade damaged mitochondrial proteins, essentially preserving the mitochondria's function and preventing a complete breakdown. What's even more fascinating is that mild chronic activation of UPRMT has been linked to increased lifespan in various animal models, suggesting it's not just a repair mechanism, but an adaptive stress response that enhances cellular resilience and overall longevity. So to synthesize this, cells possess multiple sophisticated layers of quality control, from repair responses, exemplified by UPRMT, to replacement through biogenesis, to removal via mitophagy. This deep dive prioritizes removal through mitophagy because the failure to effectively call these defective mitochondria appears to be a singularly significant contributor to the cascade of aging and its associated diseases. Mega recaps and flashcards. All
right, that was a deep dive into mitophagy itself. Let's do a mega recap. We define mitophagy as the cell's specialized cleanup crew for faulty or old mitochondria. It's crucial for maintaining a healthy power supply.
We covered the two main ways it starts: the highly studied PINK1 Parkin pathway, which tags damaged mitochondria with ubiquitin for removal, and the receptor-mediated pathways involving proteins like BNIP3 and NIX, often triggered by specific stresses like low oxygen.
And, importantly, the aging picture is nuanced. While mitophagy can decline in some tissues, it's not universal. But boosting it, like with urolithin A in mice, seems beneficial, reducing inflammation and improving function.
Exactly. And mitophagy works together with mitochondrial biogenesis, making new ones, and the UPRMT internal repair system to maintain overall mitochondrial health. We focused on mitophagy because failure here seems key to aging problems.
Perfect. Now for some flashcards, key terms from this section. Mitophagy, PINK1 Parkin pathway, E3 ubiquitin ligas, autophagy adapters like P62, OPTM, autopogosomes, lysosomes, BNIP3, NIX, FUNDC1, these are mitophagy receptors, LC3, mitochondrial biogenesis, PGC1, NRF12, TFAM, these regulate biogenesis, mitochondrial unfolded protein response, UPRMT, urolithin A.
Excellent list. Understanding these terms is key to grasping mitochondrial quality control.
Building on that concept of mitochondrial quality control, let's explore a truly fascinating and, frankly, kind of concerning phenomenon. Mitochondria aren't just isolated power generators humming along quietly in the cell's cytoplasm. What's truly critical here is that they are highly communicative organelles, actively signaling with the rest of the cell, especially the nucleus, which is the cell's command center.
That's absolutely right. And one particularly impactful and often unfortunate way they signal is by releasing what we call danger signals when their integrity is compromised. Think of it as a smoke detector going off when there's an internal fire. Damaged mitochondria, when their membranes become leaky or even rupture, can physically release their internal components. This includes mitochondrial DNA or mtDNA, which we've mentioned, and other distinct mitochondrial molecules into the cytosol, the cell's main fluid, or even in some cases, into the extracellular space outside the cell entirely. The crucial point here is that your innate immune system, the body's first line of defense, often misinterprets these leaked mitochondrial molecules. It recognizes them not as components of your own cells, but rather as indicators of an invading infection like viral or bacterial DNA or significant dangerous cell damage. These mitochondrial molecules, therefore, act as what we call DAMPs, which stands for damage-associated molecular patterns. They are essentially danger signals, directly triggering a potent inflammatory response, as if sounding a high-pitched alarm throughout the cellular neighborhood.
So if our own mitochondrial DNA leaks out, it can essentially trick our immune system into thinking we have like a viral infection. That's incredible. And there's a specific highly relevant pathway that becomes activated by these DAMPs, the CGAS STING pathway. How does that work? Can you break that down?
Indeed, the CGA STING pathway is a prime example of how these danger signals are processed. Imagine this as a molecular cascade, a series of precise steps. First, you have CGAS, which stands for cyclic GMP AMP synthase. This is an innate immune DNA sensor that normally resides in the cytosol. Its job is to be on high alert for, well, DNA where it shouldn't be. When CGAS detects DNA in the cytosol DNA that should ideally be absent or tightly contained within the nucleus or within healthy mitochondria, it's immediately triggered. Upon activation, CGAS produces a specific messenger molecule called KIBIM. The CGAMP molecule then acts as a second messenger, directly activating STING, which stands for Stimulator of Interferon Genes. STING is an adapter protein, a crucial hub in this signaling pathway. The activation of STING then leads to a robust immune response, specifically the induction of type I interferons and other pro-inflammatory cytokines. This is the initiation of a powerful cellular defense, much like the body mobilizing its immune forces. Normally, our own cellular DNA is meticulously confined to the nucleus, or in the case of mitochondrial DNA, to the mitochondria themselves. So, the presence of DNA floating freely and uncontained in the cytosol is a very strong indicator of trouble, signaling a potential viral infection, the presence of bacterial invaders, or, as we're discussing, ruptured or severely damaged organelles within the cell. And here's the crucial connection to our current topic. It has been definitively shown that mitochondrial DNA, or mtDNA, is a major activator of the CGA STING pathway. This is a profound link. A fundamental hallmark of aging, universally observed across species, is the widespread loss of mitochondrial homeostasis. This means mitochondria become less stable, more prone to dysfunction, and, critically, more likely to leak their contents. Studies in aged animals have consistently revealed that mitochondria become structurally abnormal with age. They lose their characteristic shape and internal organization, and this abnormality directly initiates the leakage of mtDNA into the cytosol. Consider a specific example. In brain immune cells, known as microglia, of old mice, researchers observed not only these misshapen dysfunctional mitochondria, but also a significant quantifiable increase in cytosolic mtDNA. This leaked mtDNA, in turn, led to the hyperactivation of the CG-Esting inflammatory signaling pathway, creating a chronic state of low-grade inflammation within the brain.
So if I'm understanding this right, this chronic low-grade inflammation, specifically driven by our own self-DNA, in this case mitochondrial DNA acting as a damp, is thought to be a key, if not central, contributor to what scientists now term inflammaging. What does inflammaging really mean for us, practically speaking?
Inflammaging is precisely the term for this smoldering systemic inflammation that is intimately associated with biological aging and contributes to a wide array of degenerative diseases. It's not the acute beneficial inflammation that helps heal a wound, but a persistent low-level fire that slowly damages tissues throughout the body. Indeed, research has shown that genetic deletion or pharmacological inhibition of STING in aged mice has remarkable effects. It significantly suppresses a broad range of inflammatory genes, especially the interferon-stimulated genes, and markedly reduces the accumulation of inflammatory cells in various tissues. This translates to tangible health benefits in these old mice, including impressive improvements in muscle endurance, enhanced strength, and even improved cognitive function. In short, an overactive CGS-STING pathway, fueled by mitochondrial damage and the leakage of their DNA, appears to be a significant, if not primary, driver of frailty and overall age-related decline. It's a fundamental mechanism linking cellular damage to systemic aging, and this is where mitophagy becomes absolutely critical again, acting as a proactive cellular defense. Mitophagy is the cell's elegant, indeed indispensable, way of preventing this harmful mtDNA leakage from occurring in the first place. By efficiently removing dysfunctional mitochondria before they can rupture and spew their contents, those danger signals like mtDNA into the cytosol, mitophagy acts as a proactive cellular defense mechanism. Recall the mitophagy reporter study we mentioned earlier. It elegantly demonstrated that old mice exhibited more interferon and inflammatory activation, specifically in tissues where mitophagy was found to be insufficient. This is direct evidence linking impaired cleanup to inflammation. Crucially, when the researchers boosted mitophagy in those old mice with urolithin A, it not only attenuated the CGA-STING activation, but, just as importantly, the animal's neurological function measurably improved. These findings strongly suggest that improving mitochondrial quality control can effectively curtail the inflammation of aging, most likely by reducing the insidious release of mitochondrial damps like mtDNA. It's a beautiful illustration of how addressing a root cause, those leaky, damaged mitochondria, can calm the downstream inflammatory consequences of inflammaging, leading to widespread systemic benefits. And beyond the CGA-STING pathway, mitochondria engage in other vital forms of mitonuclear signaling, demonstrating their role as true cellular communicators. For instance, they release reactive oxygen species, or ROS, which at lower controlled levels act as crucial signaling molecules rather than just damaging agents. We'll explore this fascinating aspect further shortly. They also subtly alter metabolic byproducts that can directly influence gene regulation. For example, the critical NADU to NADH ratio modulates the activity of sirtuins, a family of proteins deeply involved in longevity. And alpha-ketoglutarate, another mitochondrial metabolite, affects enzymes like DNA demethylases, which play a role in epigenetics. And in the most severe cases of stress, if mitochondria are terminally damaged, they can even trigger programmed cell death or apoptosis through the release of a protein called cytochrome C. So a crucial take-home from this section is that dysfunctional mitochondria behave like pervasive danger signals, forging a direct and profound link between mitochondrial dysfunction and chronic inflammation, and even contributing to the pathways of certain autoimmune diseases. This has immense implications for many diseases of aging, including severe neurodegenerative conditions like Alzheimer's and Parkinson's, as well as various cardiovascular diseases, where both mitochondrial DNA and inflammasome activation, which is another key part of the innate immune response, have been directly implicated as key players in disease progression. Therefore, controlling mitochondrial damage through robust mitophagy and constant biogenesis is not just vital for the intrinsic health and function of individual cells, it is absolutely paramount for preventing widespread collateral inflammatory damage throughout the entire body. Micro recap point 2, approximately 10 minutes.
Okay, another quick check-in. We just covered how damaged mitochondria can leak their contents, like mtDNA.
Right, and these leaked contents act as damps, damage-associated molecular patterns. They basically trigger the immune system inappropriately.
Specifically activating the CGS-STING pathway, which ramps up inflammation. This contributes to inflammaging, that chronic low-grade inflammation seen in aging.
Exactly. And crucially, efficient mitophagy acts as a defense, clearing out those leaky mitochondria before they trigger this whole inflammatory cascade. Boosting mitophagy, like with urolithin A in studies, seems to calm this inflammation.
Got it. Leaky mitochondria equals inflammation, and mitophagy is the prevention mechanism. Mega recap in flashcards. Mitonuclear signaling and inflammation. All right, let's summarize this critical link between mitochondria and inflammation. We established that mitochondria aren't isolated, they signal to the rest of the cell. When damaged, they can leak danger signals or DAMPs, particularly their own mitochondrial DNA, mtDNA.
Right. This leaked mtDNA in the cytosol is detected by the CGA-S sensor, activating the STING pathway. Think of it like an internal alarm system gone haywire. This CGA STING activation drives the production of type I interferons and other inflammatory molecules, contributing significantly to the chronic, low-grade inflammation of aging, termed inflammaging.
And this inflammaging is linked to numerous age-related diseases and frailty. But the good news is that mitophagy plays a key role in preventing this mtDNA leakage by removing damaged mitochondria before they burst.
Exactly. Enhancing mitophagy, as shown with urolithin A, can reduce this inflammatory signaling and improve function, highlighting its systemic importance. We also touched on other signaling roles of mitochondria involving ROS and metabolites.
Okay, time for the flashcards for this section. Mitonuclear signaling, damage-associated molecular patterns, DAMPs, mitochondrial DNA, mtDNA, cytosol, CGA STING pathway, CGA-CGM, STING, type I interferon, inflammaging, microglia, brain immune cells where this was observed, urolithin A, again the mitophagy booster, sirtuins, mentioned as NAD plus dependent, alpha-ketoglutarate, metabolite affecting epigenetics, cytochrome C, released during apoptosis, inflammasome, another inflammatory pathway sometimes involved. That covers the key players in this connection between mitochondrial damage and inflammation. Let's circle back to our initial altitude analogy. We envisioned hitting that wall, the body's power failing. What could have fundamentally prevented that hypothetical high-altitude collapse? What's the most practical, universally applicable intervention we have?
The answer, while seemingly simple, is profoundly powerful: training. Regular, consistent exercise stands out as one of the most potent, universally accessible, and scientifically validated ways to dramatically improve both the sheer quantity and the essential quality of our mitochondria throughout our lifespan. It is, quite literally, a mitochondrial elixir that is within everyone's reach. Endurance exercise, for instance, is a remarkably well-established stimulus for mitochondrial biogenesis, making new mitochondria. It achieves this primarily through the activation of PGC1A, often referred to as a master regulator of mitochondrial biogenesis. PGC1 orchestrates a complex symphony of gene expression that leads to the creation of new mitochondrial proteins and components. The direct result of this process is a quantifiable increase in the density of mitochondria within muscle cells. This higher density, in turn, directly enhances the muscle's oxidative capacity, its ability to efficiently use oxygen to produce ATP. This translates into an increased VO max, which is your maximal oxygen uptake, a gold standard measure of cardiorespiratory fitness. You become more efficient at utilizing oxygen to fuel your body, allowing for greater sustained effort.
But it's not just about making more mitochondria, which is already incredible. What's equally fascinating is that exercise also activates mitophagy. Think of it as a highly diligent, specialized cleanup crew that arrives, not coincidentally, after periods of exercise-induced stress. This crew's purpose is not just general tidying. It's to selectively remove and replace those weaker, potentially damaged mitochondria with the newly generated, stronger, and more efficient ones. It's a continuous process of optimization for the entire mitochondrial pool within your cells. How does exercise actually trigger this selective removal?
Let's delve into the elegant molecular mechanism behind how exercise specifically induces mitophagy. A major central player in this process is AMPK, which stands for AMP-activated protein kinase. AMPK is often referred to as the cell's primary energy sensor. Visualize this as a signaling cascade within the cell. During vigorous exercise, particularly when muscles are working hard, ATP, our cellular energy currency, is rapidly consumed. As ATP is broken down, levels of AMP, which is a breakdown product of ATP, rise significantly within the cell. This increase in AMP is the direct signal that activates AMPK. Once activated, AMPK then orchestrates a series of downstream events. It directly triggers general autophagy and, more specifically, mitophagy by phosphorylating, meaning adding a phosphate group to another crucial protein called ULK1 or UMP51 like kinase 1. ULK1 is the initiating kinase for the entire autophagy pathway. It's the first domino to fall. And notably, AMPK phosphorylates ULK1 at a specific site, serine 555, which seems crucial for targeting autophagosome, specifically to mitochondria for degradation. And we have solid proof for AMPK's requirement in this process. Studies using muscle-specific AMPK knockout mice, these are mice genetically engineered so that the AMPK gene is deleted only in their muscle tissue, have clearly shown that AMPK is, in fact, required for exercise-induced mitophagy. In these knockout mice, the usual and expected increase in autophagy and mitophagy signaling observed after periods of exercise or fasting is significantly blunted. Furthermore, as these mice age, they accumulate a higher burden of abnormal mitochondria compared to their wild-type counterparts, unequivocally highlighting AMPK's vital and indispensable role in maintaining mitochondrial quality and preventing age-related decline.
So this isn't just theory or observation in highly controlled lab settings. It translates to real-world experience, right? In both mice and humans. A single bout of exercise has been consistently observed to increase the activation of both AMPK and ULK1 in muscle tissue. This activation coincides precisely with tangible evidence of mitophagy occurring during the post-exercise recovery period. Researchers have obtained direct evidence, using those advanced reporter systems, that even a single bout of endurance exercise significantly increases mitophagy in muscle during the subsequent recovery phase. And, importantly, when ULK1 was genetically knocked out specifically in muscle, the mice were unable to properly clear damaged mitochondria after exercise. Consequently, they showed poor metabolic adaptations to training, underscoring ULK1's absolutely indispensable role in this adaptive process. So, in essence, exercise triggers a transient wave of mitophagy, effectively clearing out the junk mitochondria that may have been damaged by the exercise bout itself. This crucial cleanup then stimulates the replacement by new, more robust mitochondria during the recovery period. This entire dynamic cycle is considered fundamental to how exercise progressively improves overall mitochondrial function and resilience over time.
And it's crucial to emphasize the human relevance here. It's not just in lab mice. Human studies provide compelling and consistent support that exercise truly enhances mitochondrial quality control. For example, a significant study on older adults revealed that those who had maintained a consistent endurance training habit throughout their lives had significantly higher levels of parkin, that key mitophagy protein we discussed earlier, in their muscle tissue compared to sedentary age-matched controls. Markers of mitochondrial fission, a process that often precedes mitophagy by fragmenting older mitochondria, were also notably elevated in these active individuals. Crucially, their muscle tissue exhibited higher oxidative enzyme levels and superior respiratory capacity, directly linking the molecular changes to better energy production. In fact, the muscle of a 70-year-old lifelong endurance athlete can strikingly resemble that of a much younger individual in terms of both mitochondrial content and function, a truly remarkable testament to the power of consistent physical activity. This study concluded that lifelong physical activity is instrumental in preserving mitochondrial quality control, encompassing both fission and mitophagy and overall function despite the natural aging process. Similarly, another comprehensive analysis noted that citrate synthase activity, which is a classic and widely accepted biochemical marker of mitochondrial density, along with OXP-HOS enzyme levels, typically decline with age in sedentary people. However, consistent exercise training can powerfully counteract or significantly blunt this age-related decline. Citrate synthase is a validated biomarker of muscle mitochondrial content, and increases in its activity after training directly correlate with improvements in whole body VO2max, which, as we mentioned, is the gold standard measure of cardiorespiratory fitness. These aren't just subtle shifts, these are robust, measurable physiological improvements.
So given all this, a brief but vital note on training strategy emerges. The concept of periodized training. This involves intelligently varying the intensity, volume, and rest in structured cycles to maximize physiological adaptations and prevent plateaus or overtraining. For optimal mitochondrial health, periodization means intentionally balancing stress and recovery. High-intensity interval training, or HIA for instance, can provide potent short bursts of stimulus for both mitophagy and biogenesis, creating that acute stress that triggers adaptation. But easier sessions or dedicated rest days are equally crucial as they allow the essential rebuilding phase, the biogenesis of new robust mitochondria, to fully occur.
A word of caution here. Overtraining without adequate recovery can be counterproductive. Excessive and unrelenting stress might induce damage faster than the cellular repair mechanisms can possibly keep up, leading to a net deficit in mitochondrial health. Conversely, strategically incorporating intervals and endurance sessions, carefully spread across the week with sufficient recovery, can progressively elevate the bar for mitochondrial performance and capacity. This optimized approach yields tangible, measurable results. Improvements in VO-max, increased citrate synthase and specific complex enzyme activities in muscle, and significantly better fatigue resistance. In our altitude trek analogy from the intro, a well-structured periodized training plan leading up to the climb, ideally even including some hypoxic training if feasible, would dramatically increase mitochondrial efficiency and resilience, effectively sparing the hiker from a devastating bioenergetic crash and allowing them to summit with greater ease. Micro recap point 3, approx 15 minutes.
Okay, quick recap on exercise. We've established it's a powerhouse intervention for mitochondria.
Absolutely. It boosts both biogenesis, making new mitochondria, largely via PGC1, and mitophagy, the cleanup process.
And that cleanup, mitophagy, is triggered by AMPK, the cell's energy sensor, activating ULK1 during and after exercise.
Right. We see evidence in both animal models, like the AMPK knockout mice, and importantly, in human studies showing higher Parkin, better enzyme activity, and preserved function in active older adults.
Plus, we touched on the importance of smart training periodization, balancing stress like HIIT with recovery to maximize those mitochondrial gains.
Exactly. It's about working smarter, not just harder, to get the best results. Mega recap and flashcards. Exercise is mitochondrial elixir.
Let's synthesize the power of exercise for our mitochondria. We started by revisiting the altitude analogy, training as the key preventative measure. Exercise robustly stimulates mitochondrial biogenesis, increasing mitochondrial density and oxidative capacity, often measured by VO-max.
And critically, it also activates mitophagy, the cleanup crew. This is primarily driven by AMPK sensing energy changes during exercise, which then activates ULK1 to initiate mitochondrial removal. It's a cleanup and rebuild cycle.
We highlighted compelling human evidence. Lifelong exercisers maintain higher levels of mitophagy proteins like Parkin, preserve mitochondrial enzyme activity like citrate synthase, and maintain function much better than sedentary peers.
And we stress the importance of periodized training, strategically varying intensity, like HIIT, and volume with adequate recovery to optimize these adaptations and avoid overtraining. The measurable benefits include increased citrate synthase activity and higher VO-max.
Great. Flashcard time for the exercise section. AMPK, AMP-activated protein kinase. ULK1, UNC51 like kinase 1. PGC1, master regulator of biogenesis. Mitochondrial density. Oxidative capacity. VO-max, Parkin, the mitophagy protein observed higher in active humans. Citrate synthase, CS activity, marker of mitochondrial density. OXPHOS enzyme levels. Periodized training, high-intensity interval training, HIIT. Lactate threshold, another functional marker improved by training. That covers the essentials of how exercise reshapes our mitochondrial landscape. Let's shift our focus now to another giant in the field of aging research, a molecule that has garnered immense attention, and for very good reason, NAD. This crucial coenzyme, nicotinamide adenine dinucleotide, is at the heart of much of the discussion around cellular metabolism and longevity. What makes it so important?
NAD is indeed absolutely crucial. It plays a dual, indispensable role within the cell, particularly in the mitochondria's energy production processes. It serves as a primary carrier of electrons in what are called redox reactions. These are the fundamental chemical reactions where electrons are transferred, enabling the flow of energy that ultimately produces ATP. But NAD's importance extends far beyond just energy production. It also functions as a vital substrate for a suite of longevity-related enzymes, most notably the sirtuins and the PRPs, or poly-ADP ribose polymerases. Given its central role in both energy metabolism and cellular signaling, it is often accurately described as a fundamental currency of cellular metabolism and overall cell health. And this is where the NAD question truly begins. Levels of NAD show a clear, consistent, and widely reported tendency to decline with age. Studies conducted in both rodent models and human populations have repeatedly found significant drops, ranging from 20% to as much as 50% in NAD levels across various tissues of older animals and humans compared to their younger counterparts. This isn't a minor fluctuation. It's a substantial depletion of a vital cellular resource.
So if NAD is so central to our cellular functions, why is this age-related decline such a significant issue for cellular health and aging? What are the ripple effects?
The cascading effects of low NAD are profound and touch upon multiple critical cellular functions. First, low NAD directly impairs mitochondrial function. This is because key reactions within the TCA cycle, also known as the Krebs cycle, and the electron transport chain, the heart of mitochondrial energy production, are highly dependent on a balanced NAD ratio. Without sufficient NAD acting as an electron acceptor, energy production falters, leading to less ATP and overall cellular sluggishness. Second, it significantly hampers DNA repair. PRE enzymes, which are critical for recognizing and repairing DNA damage, consume NAD as part of their catalytic activity. Reduced NAD levels mean these essential DNA repair processes become less efficient, leading to an accelerated accumulation of DNA damage over time. And third, it reduces sirtuin activity. Sirtuins are a family of proteins that are deeply implicated in longevity, metabolic regulation, and even influencing mitochondrial biogenesis. These are NAD-dependent enzymes, meaning they absolutely require NAD to function. A drop in NAD levels directly hinders their activity, impacting a wide array of protective and metabolic pathways. Thus, the NAD question is whether strategically boosting NADO in older individuals can effectively restore a more youthful metabolic state and enhance cellular resilience, thereby mitigating aspects of age-related decline. One major prominent reason identified for this pervasive NADO decline with age is the enzyme CD38. CD38 is a specific type of enzyme known as NADO. Its primary physiological role, quite literally, is to consume NADO, breaking it down in the process producing other signaling molecules like cyclic ADPR. CD38 is highly expressed on the surface of various inflammatory cells, such as macrophages and certain lymphocytes, which are key components of your immune system, as well as on endothelial cells that line our blood vessels. Over many years of dedicated research, Dr. Eduardo Chini and his colleagues have consistently shown that CD38 activity significantly increases with age. This increased activity positions CD38 as a key driver of NAD loss during the aging process, acting like an insatiable drain on the cellular NADO pool. In aged mice, for instance, elevated CD38 expression leads to accelerated NADO breakdown and contributes directly to mitochondrial dysfunction, partly by reducing the activity of SIRE3, a specific mitochondrial sirtuin that, like all sirtuins, requires NADO to function optimally. In fact, compelling proof of CD38's role comes from experiments where genetically deleting CD38 or pharmacologically inhibiting it in mice can effectively raise their NADO levels and, remarkably, improve various aspects of their metabolic health.
So, what exactly causes CD38 to ramp up its activity so significantly in old age? What's the deeper, perhaps more insidious mechanism at play here that connects into the aging process itself?
That's a profound question, and it unveils a fascinating, rather insidious, vicious cycle. It turns out that senescent cells, those often referred to zombie cells that have stopped dividing but remain metabolically active and, crucially, secrete harmful factors, play a critical role. These senescent cells secrete a complex cocktail of pro-inflammatory factors collectively known as the SASP, which stands for senescence-associated secretory phenotype. This SAP is a potent driver of chronic inflammation. A pivotal 2019 study shed a crucial light on this. It revealed that senescent cells themselves did not necessarily have higher CD38 levels. However, the SASP cocktail of cytokines and chemokines that they secrete caused other cells in the immediate microenvironment, specifically immune cells like macrophages, to greatly increase their CD38 expression and activity. Here's how this vicious cycle unfolds step by step, like a detrimental feedback loop. First, with advancing age, more and more cells in your body become senescent and begin to secrete these inflammatory SASP factors. Second, these SSP factors act as molecular distress signals, recruiting and altering other immune cells, essentially activating them and causing them to turn on and upregulate CD38 expression. Third, the activated CD38 enzymes now abundant in these tissues rapidly chew up and deplete the vital NAD pool. And finally, low NAD in turn can impair various cellular functions, including critical mitochondrial maintenance, DNA repair, and sirtuin activity. This impairment can potentially promote even more cellular dysfunction and indeed more senescence, thereby perpetuating and amplifying the entire cycle. As one paper powerfully summarized, SASP-mediated upregulation of CD38 can disrupt cellular NAD homeostasis, establishing a direct and critical link between cellular senescence, chronic inflammation, and the age-related decline in NADU. In essence, chronic inflammation or inflammaging and NADU metabolism are intricately intertwined. CD38 immune cells, activated by SSP-driven inflammation, actively drain the essential NAD pool from tissues, creating a systemic deficiency. Now, while CD38 is a major player, another contributor to NADU decline is the increased activity of PRP enzymes. PRPs consume NAD to repair DNA, and their activity naturally ramps up in aging due to the accumulation of DNA damage over a lifetime. However, CD38 is generally considered the dominant NAD sink in many tissues, although its precise role in the brain may be less clear and requires further research. All of this matters immensely because NAD is absolutely required for mitochondrial sirtuins, particularly SIRT3. These sirtuins are crucial for maintaining mitochondrial protein health and robust antioxidant defenses within the mitochondria themselves. Therefore, consistently low NAD levels can significantly exacerbate mitochondrial oxidative stress and overall dysfunction, creating a perfect storm platform for cellular decline, where the very energy-producing organelles are both inefficient and vulnerable. It's a complex interplay where aging leads to increased NAD consumption, primarily by CD38 and PARPs, and in some cases, even reduced NAD production, as some enzymes in the NAD salvage pathway also decline. This leaves our mitochondria energy-starved and our cells increasingly vulnerable to accumulating damage, contributing to the systemic frailties of aging. Mega recap and flashcards. The NAD question.
Okay, let's recap this crucial NADO story. NAD is a vital coenzyme for energy, redox reactions, and a substrate for key enzymes like sirtuins and PRPs. It's fundamental to cell health.
And the big issue is that NAD levels consistently decline with age, sometimes by up to 50%. This decline is problematic because it impairs mitochondrial function, hampers DNA repair via PRPs, and reduces the activity of protective sirtuins.
A major reason for this decline seems to be the enzyme CD38, a netidase whose activity increases with age. This increase is driven in part by a vicious cycle involving senescent cells.
Right. Senescent cells release inflammatory SASP factors, which cause immune cells to express more CD38. This CD38 then chews up NAD, potentially worsening dysfunction and creating more senescence. PRP activity also contributes to NAD depletion due to increased DNA damage.
And this loss of NAD is especially bad for mitochondria because key protective enzymes inside them, like SIRT3, absolutely require NAD to function.
Exactly. It creates that perfect storm scenario for cellular decline. Now for the flashcards. Angdara-U, nicotinamide adenine dinucleotide, sirtuins, PARPs, poly-ADP ribose polymerases, CD38, anases, Dr. Eduardo Chini, key researcher in CD30 field, cyclic ADPR, signaling molecule produced by CD38, senescent cells, zombie cells, SAHES, a senescence-associated secretory phenotype, SIRT3, key mitochondrial sirtuin, DNA damage, NAD salvage pathway, how NAD plus is made recycled.
Great summary. That sets the stage perfectly for discussing NAD boosters. Given NAD's profound and undeniable importance in cellular function, it's really no surprise that a wave of supplements and compounds have emerged, aiming to boost NAD levels as a potential strategy to improve health and optimistically slow aspects of aging. It's a hugely attractive proposition, right?
Yeah, absolutely. And the two most popular compounds in this space are nicotinamide riboside, NR, and nicotinamide mononucleotide, NMN. Both of these are derivatives of vitamin B3, and they serve as NAD precursors. This means they are the raw building blocks, the molecular components that the body can use to synthesize NAD. When taken orally, these compounds are absorbed, enter cells, and are then converted into NAD via the salvage pathway, which is a crucial metabolic route for efficiently recycling and synthesizing NAD within the body. The initial excitement around these compounds stemmed from numerous preclinical studies conducted across a variety of model organisms, including worms, flies, and mice. In these studies, giving NR or NMN consistently demonstrated remarkably impressive benefits. These benefits included improved mitochondrial function, enhanced insulin sensitivity, meaning cells responded better to insulin, which is crucial for metabolic health reductions in neurodegeneration in various disease models, and even in some cases extended lifespan, particularly observed in sick mice or those on high-fat diets. Such compelling and often dramatic results in animal models rapidly generated immense excitement within both the scientific community and the public, leading to the rapid initiation of many human clinical trials.
Okay, so the big question then becomes, are those profound effects consistently observed in animal models truly translating into robust clinical signals and tangible health benefits in human beings? That's the leap of scientific translation that everyone is watching. Where do we stand on that now?
And the nuanced answer so far is, the evidence is only partially conclusive, with results often being mixed and, frankly, quite modest in human trials compared to the dramatic effects seen in preclinical models. First, let's address the good news on the biochemical efficacy side. Multiple human trials have consistently and unequivocally confirmed that taking NR or NMN does indeed increase NAD levels in the blood and, importantly, in some accessible tissues of humans. This is a confirmed biochemical effect. Furthermore, supplementation with both NR and NMN is generally considered safe and well tolerated across various studies, with no serious adverse effects reported even at relatively high doses, for example, up to 2,000 milligrams per day. Indeed, these supplements effectively boost NAD abundance, with some studies showing a significant increase, often around 60% to 100% in blood NAD metabolites after just a few weeks of consistent NR or NMN intake. So the good news is that these supplements work in the fundamental sense of elevating the desired molecule, NAD, and they appear to be safe in the short term. However, the real challenge lies in the clinical efficacy, meaning actual, tangible, and consistent improvements in health outcomes or functional measures. This aspect remains largely unclear. A recent comprehensive review that aggregated data from various human trials summarized the situation pointedly. The clinical evidence that raising NADO concentrations can improve physiological function is unclear. Many of the human trials conducted to date have been characterized by relatively small sample sizes and short durations, often making it inherently difficult to detect subtle or long-term functional changes that might accumulate over a longer period. The results, as mentioned, have been a mixed bag. For instance, we've seen null findings in critical metabolic parameters. In studies involving healthy middle-aged and older adults, NR has not consistently improved key metabolic markers. A 2018 randomized trial involving obese men, for example, found that 12 weeks of NR at a dose of 1,000 milligrams per day did reliably raise NAD levels, but did not significantly improve insulin sensitivity or glucose metabolism when compared to a placebo group. Similarly, this specific trial observed no major changes in muscle mitochondrial function or inflammatory markers, despite the clear biochemical effect of increased NADO. Conversely, some trials do report mild benefits. One study in older adults noted that NR slightly lowered blood pressure in individuals who already had hypertension and showed some modest improvement in arterial stiffness, which is a marker of cardiovascular health. Another pilot study found that NR improved muscle NADO levels and reduced levels of liver fat in people with non-alcoholic fatty liver disease. However, it's crucial to note that these positive effects, while encouraging, have generally been modest in magnitude, not the dramatic fountain of youth type effects often observed in animal models. NMN trials, which began a bit later due to its more recent availability as a supplement, also consistently demonstrate increased NADO levels and offer some intriguing hints of clinical benefit. For example, a 2021 trial found that NMN improved muscle insulin sensitivity, specifically in pre-diabetic older women. And a 2022 study involving recreational runners found that NMN slightly increased their muscle oxygen uptake and aerobic capacity during exercise. However, other NMN studies showed no significant effect on muscle strength or overall performance beyond what was seen with placebo, again illustrating the mixed nature of the results. So, why these mixed results, and what's the future outlook? Overall, a key limiting factor is that sample sizes have been generally small, and often the studies are simply not long enough to adequately detect meaningful or statistically significant functional changes that might take longer to manifest. The comprehensive review of NAD boosters in humans also highlights that dosing regimens vary widely across studies, and many studies may be statistically underpowered to detect anything beyond the initial NADU increase. It's also entirely plausible that NAD restoration does help, but perhaps only in very specific conditions or in particular subpopulations. For example, individuals who are already under significant metabolic stress or those with clearly diagnosed NAD deficits might show a more robust response. In generally healthy individuals, simply flooding cells with more NAD cursors might result in any excess NAD being rapidly broken down or excreted without providing a significant performance boost beyond their already healthy baseline.
Therefore, the bottom line on NR and NMN is becoming clearer. They reliably raise NAD levels. This is a consistently confirmed biochemical effect across multiple studies. However, whether they consistently produce tangible clinical benefits, such as increased energy, enhanced endurance, effective disease prevention, or significant improvements in overall health span, is still not definitively proven. The signals are decidedly mixed with some positive findings, but also a significant number of null findings. This means we have a lot more to learn and much more research is needed. It's a classic illustration of the inherent challenges in translating exciting findings from the bench, the controlled environment of laboratory research to the bedside, the complex reality of clinical application in humans. Dramatic effects often observed in animal models do not always directly translate with the same magnitude to human physiology, especially if human studies are not meticulously designed to target the most relevant outcomes or specific patient populations. Looking ahead, ongoing larger and more rigorously designed human trials, including one testing NMN for muscle weakness in older adults and another testing NR for cognitive aging, are crucial and will undoubtedly shed much more definitive light on these compounds. Micro recap point 4, ProBooks 20 minutes. Okay, quick recap on NAD boosters like NR and NMN. They're precursors the body uses to make NAD plus acyl.
Right. Animal studies showed really impressive benefits, sparking lots of excitement in human trials.
But the human results are mixed. They definitely do raise NAD plus levels biochemically and seem safe short-term.
Exactly. But the clinical benefits, actual improvements in function like metabolism, endurance, strength are much less clear. Some studies show mild effects, many show none.
So the hype might be ahead of the solid human evidence right now. We need more, bigger, longer studies to know for sure.
That's the current state of play. Biochemically effective, clinically uncertain. Mega recap and flashcards. NAD boosters, NR, and N and MN.
Let's wrap up this section on NAD boosters. There's widespread interest in compounds like nicotinamide riboside and NR and nicotinamide mononucleotide and MN. They function as precursors, providing the raw materials for our cells to make more NAD via the salvage pathway.
The excitement largely stemmed from impressive preclinical data in animals, showing benefits for mitochondria, metabolism, and even lifespan in some models. This led to numerous human clinical trials. However, the results in humans have been mixed.
What's clear is the biochemical effect. NR and NMN do reliably increase NAD levels in humans and are generally safe. That part seems solid.
Yes, but the clinical efficacy, consistent, tangible improvements in function or health outcomes remains uncertain. We've seen some null results, particularly for metabolic benefits in healthy older adults, alongside some reports of modest improvements in things like blood pressure or muscle insulin sensitivity in specific groups.
So the current verdict is biochemically proven, clinically mixed, and modest. It underscores the challenge of translating animal findings to humans and the need for larger, longer, more rigorous trials.
Precisely. Now for the flashcards. Nicotinamide riboside, NR, nicotinamide mononucleotide, NMN, NADG precursors, salvage pathway for NAD plus synthesis recycling, preclinical studies, animal models, clinical efficacy, actual health benefits in humans, insulin sensitivity, arterial stiffness, muscle oxygen uptake, aerobic capacity.
Good list. That covers the key players and concepts in the NAD booster debate.
Rather than solely focusing on the strategy of pouring in more NAD precursors with supplements, another highly promising and arguably more elegant strategy is to focus on blocking NAD consumption. The logic here is quite compelling. Since CD38 has been unequivocally identified as a major NAD2 consumer and a key driver of its decline in aging, inhibiting this enzyme has gained significant interest as a more upstream way to effectively preserve and raise NAD levels. This approach is currently still predominantly in preclinical stages, meaning there are no approved CD38 inhibitors specifically for NAD boosting in humans yet, apart from CD38 blocking antibodies that are already used in certain cancer therapies to target cancerous cells expressing CD38. However, the experiments conducted in mice using this approach are undeniably promising and provide strong proof of concept. Let me detail a pivotal study in this area. Researchers at Mayo Clinic, collaborating with a biotech company, developed a potent small molecule CD38 inhibitor designated as 78C. They administered the CD38 inhibitor to naturally aged mice, commencing the treatment at approximately 1 year of age, which is equivalent to middle age in humans, and continued the treatment for about 10 months. The results were truly striking. The treated old mice lived longer and notably healthier than their control counterparts who did not receive the inhibitor. Specifically, their median lifespan increased by approximately 10% in the 78C treated group, a significant extension translating to about 3 to 4 extra months of life for the mice. Even more compellingly, various measures of health span significantly improved. These mice exhibited enhanced exercise capacity and endurance, meaning they could run longer on a treadmill. They also showed improved grip strength, which is a clear indicator of better muscle function. Furthermore, they demonstrated improved metabolic function in late life, including better glucose tolerance. These substantial and multifaceted benefits were consistently observed in both male and female mice, although with some sex-specific differences in the degree of improvement. This study provided the first direct and compelling evidence that targeting CD38 can indeed extend both lifespan and health span in a normal aging model, not just in specific disease models. The underlying mechanism is believed to be that by blocking CD38, these mice were able to maintain significantly higher NAD levels across various tissues, which in turn preserved mitochondrial function and overall tissue health, leading to a demonstrable delay in frailty and mortality. Other research teams are also actively exploring various CD38 inhibitors or blockers using different approaches. There's even an experimental vaccine against CD38 being tested in mice. The goal of this vaccine is to stimulate the body to produce antibodies that specifically neutralize CD38 activity. One study reported that this innovative vaccine approach improved metabolic health in old mice and effectively reduced inflammation, hinting at yet another viable therapeutic route for targeting CD38. While natural compounds like apigenin and quercetin have been shown to inhibit CD38 in test tubes in vitro experiments, their potency and specificity in vivo within a living organism are generally low. More specific and drug-like small molecules are actively in development, aiming for higher efficacy and safety.
So what's so fascinating here is, why is CD38 such a uniquely compelling target compared to simply boosting NADO with precursors? It feels like it hits two crucial areas at once, inflammation and NADO. Is that the idea?
You've hit on the precise reason for the high interest. The appeal of CD38 as a target is profound because it links not only to NAD metabolism, but also intimately to immune modulation. CD38 itself is an important activation marker found on immune cells. In aging, there's a noticeable and problematic rise in CD38 inflammatory cells, particularly certain types of macrophages that some researchers describe as metabolically hyperactive. These cells contribute significantly to widespread tissue inflammation, and crucially, they are actively depleting the NADA pool. Stemming this tide with a CD38 inhibitor presents a tantalizing possibility to kill two birds with one stone, simultaneously reduce chronic inflammation, which contributes to inflammaging, and boost NADU levels by preventing its rapid degradation. Indeed, the 78C treated mice in the study also exhibited lower levels of various inflammatory cytokines, which is signaling molecules that promote inflammation. This finding is entirely consistent with the hypothesis that higher NAD levels, or perhaps the direct abscess of excessive CD38 activity, dampen age-related inflammation, contributing to the observed health benefits. Of course, it's crucial to provide a balanced perspective. CD38 does have normal beneficial physiological functions, particularly in certain aspects of immune responses. Therefore, long-term inhibition of CD38 needs to be approached with careful consideration and rigorous, comprehensive safety studies to understand any potential off-target effects. Nonetheless, these compelling preclinical findings firmly position CD38 as a highly promising target to mitigate various aspects of age-related decline. An exciting hypothesis emerging from this work is that if you can effectively fix the upstream leak in the NAD bucket, that is by inhibiting excess CD38 activity, then adding NAD precursors like NR or NMN might work far more effectively because the newly produced NAD won't be instantly consumed and wasted. Some researchers even propose a synergistic approach, combining a low-dose CD38 inhibitor with a low-dose NAD precursor to achieve more robust and sustained elevation of NAD levels than either intervention alone. As of 2025, no human trials have yet tested CD38 inhibition, specifically for NADU enhancement, but it remains a crucial space to watch closely for future developments, representing a significant upstream lever in the aging process. Mega recap and flashcards. Targeting CD38.
Okay, let's recap the CD38 strategy. Instead of just adding NAD plus precursors, this focuses on blocking NAD plus consumption.
Exactly. Since the enzyme CD38 is a major consumer of NAD plus and increases with age, inhibiting it is a promising upstream approach. It's mostly preclinical right now, but the data is exciting.
You highlighted the 78C inhibitor study in mice that showed blocking CD38 extended both lifespan and health span, improving exercise capacity, grip strength, and metabolism.
Right, a really significant finding. Other approaches like CD38 vaccines are also being explored. The high interest comes from CD38 linking both NAD plus metabolism and immune modulation inflammation.
So inhibiting CD38 could potentially tackle inflammation and boost NAD plus, simultaneously killing two birds with one stone, though safety needs careful study.
Precisely. It's a compelling target. Flashcard time. CD38 inhibitor. 78C, the specific small molecule CD38 inhibitor studied, median lifespan, health span, exercise capacity, grip strength, metabolic function, CD38 vaccine, apigenin, natural compound weak inhibitor, quercetin, natural compound weak inhibitor, inflammatory cytokines reduced by CD38 inhibition, immune modulation, CD38's role here.
Got it. A potentially powerful upstream lever. Beyond NAD boosters, which has certainly captured a lot of attention, several other metabolites have recently been highlighted as potential interventions for aging. It feels like new possibilities are emerging all the time, sometimes with a fair bit of hype attached.
Indeed. Two particularly notable ones that have generated significant discussion are taurine and alpha-ketoglutarate, or AKG. It's important to flag these as hypothesis-level compounds at this stage. This means they are largely still in the research and hypothesis generation phase, unlike, for example, exercise, which is a thoroughly proven intervention for health and longevity. We'll briefly examine what is known about them, focusing on the evidence versus the burgeoning hype, and how they compare to tried-and-true methods like physical training. Let's start with taurine. This is an amino acid-like nutrient. A headline-grabbing study published in the prestigious journal Science in mid-2023 by Dr. Vijay Yadav and his team garnered considerable attention across the scientific community and beyond. They reported a consistent and significant finding. Taurine levels demonstrably decline with age in both animals and humans. This established an intriguing correlation. More remarkably, their study showed that supplementing taurine in middle-aged animals demonstrated a profound extension of both their lifespan and their health span. To put this into context, giving a daily taurine supplement to mice from an age equivalent to about 45 human equivalent years onward resulted in a measurable increase in average lifespan by approximately 10% to 12%. For these mice, this translated to about 3 to 4 extra months of life, which is substantial in their relatively short lifespans. Even more impressively, these taurine-fed mice were healthier in old age than their control counterparts. They gained less weight, exhibited greater energy expenditure, showed increased bone mass, possessed better muscle strength and endurance, improved their glucose tolerance, and notably displayed a more youthful immune profile. The study wasn't limited to mice. It also tested taurine in other species, showing it extended lifespan in worms. In rhesus monkeys, a 6-month taurine supplementation regimen improved important physiological markers like bone density, blood glucose control, and liver health, further suggesting broad-spectrum benefits. At the cellular level, taurine appeared to act by reducing cellular senescence, that is, fewer of those problematic zombie cells, improving mitochondrial function, and reducing DNA damage in various tissues. The authors of this landmark study posited a compelling hypothesis that taurine deficiency itself might be a direct driver of aging, given the striking observation that older individuals have significantly lower taurine levels in their blood than younger ones. One specific analysis found that 60-year-olds had only about 1/3 the taurine levels of 5-year-olds, a dramatic difference. They even found, in a large cohort of 12,000 human participants, that higher circulating taurine levels correlated with indicators of better health, including less obesity, diabetes, and hypertension.
That's certainly compelling data. But what are the caveats here before anyone starts rushing to supplement? Is robust human clinical evidence still lacking? Or is this ready for prime time?
You're right to ask. While this is undoubtedly exciting data, it's vital to introduce some crucial caveats. Robust human clinical evidence is still lacking. A compelling hypothesis, no matter how intriguing or well supported by animal data, is not yet a proven therapy in humans. The proposed mechanisms are still somewhat speculative, with the notion that taurine might act as a mitochondria-friendly factor involved in mitochondrial protein folding and membrane stabilization, and also as an inhibitor of inflammation and senescence. And importantly, it's highly notable that exercise naturally raises taurine levels. The study found that after a strenuous workout, people's taurine levels significantly increased, hinting strongly that some of the profound benefits of exercise might actually be partially mediated by taurine. As of now, human trials are indeed being planned to investigate whether taurine supplementation can truly improve functional outcomes in humans. Until those rigorous trials are completed and replicated, taurine remains a promising hypothesis. It is widely found in many energy drinks and common foods, and is also produced endogenously by our bodies, making supplementation feasible and generally considered safe at moderate levels. However, we desperately need confirmation of whether it truly is an elixir of life in people or just an interesting clue from animal models before making any definitive claims. Moving on to alpha-ketoglutarate, or AKG. AKG is a crucial metabolite in the Krebs cycle, also known as the TCA cycle, which operates within mitochondria, and it also plays a role in amino acid metabolism. Similar to NADU and taurine, AKG levels tend to decline in the blood with age. A few years ago, specifically in 2020, researchers from the Buck Institute for Research on Aging, along with a startup company, investigated AKG supplementation in mice. They used a stable form of AKG called calcium AKG, which was simply added to the diet of middle-aged mice. The results were published and showed that late-life AKG supplementation could extend median lifespan by approximately 5% to 10%, with significant effects observed in female mice and a clear, albeit smaller, trend in males. More notably, similar to taurine, AKG-fed mice remained healthier for longer. They exhibited better fur condition, improved gait, and a significantly lower incidence of various chronic diseases in old age, indicating an improvement in healthspan. AKG also appeared to reduce systemic inflammation, as treated mice had lower levels of pro-inflammatory cytokines and, conversely, higher levels of IL-10, which is a key anti-inflammatory cytokine. The authors described a remarkable compression of morbidity, meaning the AKG mice not only lived a bit longer but spent a greater proportion of their extended lives in good health, effectively delaying the onset of age-related diseases and improving quality of life in later years. Mechanistically, AKG might influence aging through multiple pathways. It can inhibit the mTOR pathway, which is a major nutrient sensor intricately involved in regulating aging and cellular growth. It serves as a substrate for enzymes that affect the epigenome, potentially influencing stem cell aging and function, and its robust anti-inflammatory effect could significantly slow down the broader pro-aging cascade. There's also evidence that AKG helps preserve stem cell function and bone mass, as AKG supplementation increased bone strength in aged mice.
It's interesting to note that AKG is already marketed as a supplement, often combined with vitamins. For instance, there's a product called Rejuvant that sensationally claims an 8-year reduction in biological age, which, as you mentioned, is based on a small, non-controlled study. What's the realistic assessment for human benefits here? Should we be cautious?
And that's precisely why it is crucial to view such claims with significant caution. The mouse data on AKG's modest extension of lifespan and healthspan is considered solid and intriguing, but human data remains very sparse and preliminary. AKG is naturally produced in the body, particularly during exercise or fasting when AKG levels rise, and it is generally regarded as safe as a supplement at moderate doses. However, the key unanswered question is whether simply taking it in supplement form confers any real, measurable benefit for an already healthy human who might not have a specific deficiency. Some human trials are commencing, for example, testing AKG in osteoporosis or frailty. At this point, AKG remains at the hypothesis level. It presents a potentially exciting mechanism reducing chronic inflammation and frailty via a key metabolite, but it absolutely demands rigorous, independent human testing before widespread adoption. It is highly telling that both taurine and AKG are intimately linked to exercise. As we mentioned, exercise acutely increases taurine levels, and fasting and exercise both elevate AKG levels. Many scientists now hypothesize that these metabolites might actually be some of the crucial mediators through which lifestyle interventions, particularly regular physical exercise, exert their profound and well-established health benefits. However, the critical unanswered question remains. If one simply takes the pill without engaging in the exercise, will you achieve the same magnitude of benefit? That is currently unclear and highly doubtful. Importantly, the sheer magnitude of positive effect produced by regular, consistent exercise dwarfs what any single pill or supplement has proven so far in humans. We know unequivocally that training can increase VO max, your maximal oxygen uptake, by 15% to 30% within just a few months, representing a major functional gain in cardiorespiratory fitness. It also significantly improves muscle strength, enhances insulin sensitivity, boosts mood, and triggers a cascade of systemic benefits across virtually every organ system. No supplement to date, including NAD boosters, taurine, or AKG, can credibly claim such broad, robust, and consistently replicated effects in human beings. Therefore, these emerging compounds may eventually become valuable adjuncts in a comprehensive health strategy. For example, an older athlete might consider taking AKG to potentially aid recovery or reduce inflammation in conjunction with their training. But they absolutely will not replace the foundational, proven benefits of consistent exercise, sound nutrition, and adequate sleep for comprehensive mitochondrial health and overall longevity.
So, to provide a concise summary on these emerging compounds, taurine and AKG are undeniably intriguing, and the animal studies supporting their potential are compelling. But in humans, we should currently consider them as experimental, until rigorous, large-scale clinical trials demonstrate otherwise. They highlight important biological pathways, such as mitochondrial unfolded protein maintenance and anti-inflammatory metabolism, pathways that we can already effectively activate through existing proven means, like regular exercise or strategic intermittent fasting. It's a recurring theme that we observe in longevity science. Many breakthrough compounds often seem to mimic the beneficial effects already observed from well-established lifestyle interventions, such as exercise or caloric restriction. Rather than an either-or scenario, the future might indeed involve combining strategic lifestyle choices with targeted compounds for synergistic benefits, creating a more powerful overall effect. However, the lifestyle components, particularly structured training adaptations, remain the foundational cornerstone because they are already proven, accessible, and deliver consistently measurable outcomes like increased endurance, strength, and metabolic flexibility. Mega recap and flashcards. Emerging compounds, taurine and AKG. Okay, let's recap taurine and AKG. These are metabolites whose levels decline with age, and supplementing them showed lifespan and healthspan benefits in animal studies.
Right. Taurine supplementation in mice improved lifespan, reduced senescence, boosted mitochondrial function, and correlated with better health markers in humans. AKG supplementation in mice also extended lifespan, reduced inflammation, boosting IL-10, and improved healthspan, leading to a compression of morbidity.
The proposed mechanisms are interesting for taurine, maybe mitochondrial support and less senescence. For AKG, perhaps mTOR inhibition, epigenetic effects, and anti-inflammatory action.
Yes, but, and this is a big but robust human data, is still lacking for both. They are currently hypothesis level. We need human trials to confirm any real benefits.
And crucially, both taurine and AKG levels are naturally increased by exercise. So taking them as pills might just be mimicking a small part of what exercise already does, likely much more effectively.
Exactly. Exercise remains the proven foundation, delivering far broader and larger benefits than any of these compounds have shown in humans so far. They might become adjuncts, but not replacements. Flashcard time. Taurine. Dr. Vijay Yadav led the major taurine study. Alpha-ketoglutarate, AKG. Calcium AKG, the form often used in studies. mTOR pathway, inhibited by AKG. Epigenome, potentially influenced by AKG. IL-10, anti-inflammatory cytokine increased by AKG. Compression of morbidity, healthier aging period. Rejuvent, example AKG product, claims need caution. Mediators of exercise, taurine AKG, might be these. VO-max as a robust outcome measure of exercise.
Good summary. Intriguing leads, but lifestyle remains king for now. Let's transition to a truly crucial nuance, one that ties together several threads of our discussion. We've talked a lot about mitochondria producing reactive oxygen species, or ROS, often painting them as the bad guys, the damaging byproducts of cellular metabolism. Now let's bring those concepts together with a surprising twist that might challenge some conventional wisdom about, say, antioxidants.
That's right. ROS, such as superoxide, are indeed frequently labeled as the primary culprits causing oxidative damage and contributing significantly to the aging process. And it is true that mitochondria do naturally produce ROS as a byproduct of burning fuel through their complex metabolic processes, specifically within the electron transport chain. And it is unequivocally true that high, uncontrolled levels of ROS can indeed damage critical cellular components like DNA, proteins, and lipids, thereby contributing directly to cellular aging and disease. However, there is a profound and critical nuance here, what we might call the Goldilocks principle of ROS. While high levels are harmful, low or transient bursts of ROS can actually be profoundly beneficial. Instead of solely causing damage, these controlled intermittent bursts act as vital signaling molecules that trigger and significantly ramp up the cell's inherent adaptive stress responses. This concept is formally known as mitohormesis, which means mitochondrial hormesis. Hormesis itself is the idea that a modest transient amount of stress, in this case, from mitochondrial activity in the ROS it produces, acts as a constructive signal, triggering an adaptive improvement in the cellular system, fundamentally making it more robust, resilient, and better able to handle future, potentially greater stressors. It's like a finely tuned training stimulus for your cells.
Exercise is perhaps the most classic and powerful real-world example of mitohormesis in action. When we exercise, especially during intense bouts, our muscle mitochondria consume vastly more oxygen and, as a direct result, transiently emit more ROS than they do at rest. If you were to immediately measure oxidative stress markers after a hard workout, they would indeed show a temporary increase. But this burst of ROS isn't merely damage. It's like a strategic, urgent message sent directly to the cell's central command. We're under a significant metabolic challenge here. Please initiate a robust and comprehensive adaptive response.
Exactly. The cellular response to this message is incredibly adaptive and sophisticated. The body upregulates its own endogenous antioxidant defenses. This means it increases the production of its own powerful antioxidant enzymes, such as catalase and glutathione peroxidase, which are far more potent and precisely targeted than any supplement. Simultaneously, crucial pathways for mitochondrial biogenesis and repair, such as those governed by NRF2 and PGC1, are activated. The net long-term effect of this entire cycle, the stress, the signal, and the adaptation, is the significant, enduring improvement in overall metabolic health and an enhanced stress resistance after the recovery period. This is mitohormesis in its purest form. A short-term, controlled oxidative stress leading to substantial, long-term physiological benefits. In fact, groundbreaking research has shown that completely blocking ROS during exercise can actually impair these beneficial adaptations. This is a highly counterintuitive finding for those who believe more antioxidants are always better. In a landmark study, individuals who took high doses of antioxidant vitamins, specifically vitamin C and E, were observed to have blunted increases in insulin sensitivity. This is a key metabolic benefit that is normally robustly seen with consistent exercise training. The presumed explanation is that the pharmacological doses of these antioxidants quenched the ROS so effectively, so thoroughly, that the crucial signaling necessary for cellular adaptation was significantly dampened or even entirely eliminated. A review specifically focused on mitohormesis reinforced this, noting that low, non-cytotoxic concentrations of ROS serve as essential signals to improve metabolic health, whereas, conversely, chronically high ROS levels, such as from chronic inflammation or excessive damage, are indeed harmful. It's a delicate balance.
So to put it simply, with an analogy, ROS dose and timing are paramount. You want those strategic mini fire drills, the controlled ROS bursts, to effectively train and strengthen your cellular fire department, making it more efficient and responsive. What you absolutely don't want is an unceasing, uncontrolled blaze of chronic ROS, which would simply overwhelm the system. This principle has tangible impacts, especially for athletes. Taking antioxidant pills in excess, especially around the time of exercise, can significantly mitigate exercise-induced ROS responses and potentially reduce the very stimulus your body needs to adapt and become stronger. It has been observed that athletes who megadose antioxidants sometimes experience less improvement in crucial mitochondrial enzymes and overall endurance performance compared to those who do not supplement, because they're dampening the very signals of adaptation. The same caution applies to other hormetic stressors, such as the transient stress created by sauna use or cold exposure. These also create transient oxidative stress that triggers adaptation. Artificially blocking this stress might reduce the desired hormetic effect and the long-term benefits.
Now, this doesn't imply that antioxidants are inherently bad. It means that more is not always better, and context is absolutely key. Dietary antioxidants, for instance, those found naturally in whole fruits and vegetables, are generally mild in their effects. Crucially, they come packaged with a complex array of other beneficial phytonutrients, and they interact synergistically with your body's own finely tuned endogenous antioxidant network, which is remarkably sophisticated. Problems typically arise with pharmacological high doses of single isolated antioxidants taken in pill form, which can disrupt this delicate balance. As a general piece of advice, unless an individual has a diagnosed deficiency or a specific medical need, it is often counterproductive to drown oneself in high-dose antioxidant vitamins with the expectation of anti-aging benefits. Some studies even cautiously suggest that long-term overuse of antioxidant supplements could potentially increase overall mortality or even cancer risk, possibly by impairing normal beneficial ROS signaling within cells that is necessary for proper cellular function and surveillance. Furthermore, mitochondrial ROS also play vital roles beyond just damage and signaling for adaptation. They are intricately involved in immune defense, actively helping to kill pathogens and mount effective immune responses. They also participate in broader cell signaling for crucial processes like autophagy. Therefore, the ultimate goal in managing ROS is achieving balance. First, to support your mitochondria so they are not overproducing ROS at rest, which is indicative of severe damage or inefficiency. And second, to allow them to produce the crucial physiological bursts of ROS that serve to keep the cellular defense mechanisms alert and responsively trained. Interestingly, and quite powerfully, exercise training actually reduces baseline or resting oxidative stress in the long run. Trained mitochondria, being more efficient, leak fewer electrons and thus make less ROS during rest. And concurrently, the body's endogenous antioxidant capacity is significantly higher due to the adaptations induced by training. This is a powerful win-win. Lower chronic ROS, but a retained and enhanced ability to produce acute, beneficial ROS signals during each workout, which are then effectively handled by robust, pre-primed antioxidant systems. This sophisticated mechanism may be one significant reason why exercise is broadly anti-aging and demonstrably reduces the risk of numerous diseases.
So, in practical terms, a clear warning. Beware of antioxidant pitfalls. If you're taking NAD boosters or other promising supplements with the intention of improving your cellular health, and simultaneously consuming large quantities of isolated antioxidants with the intention of preventing aging or fighting free radicals, you might inadvertently negate some of the very adaptive benefits you are seeking to achieve. For instance, someone taking NR to boost mitochondrial function should allow those mitochondria to experience a healthy, transient oxidative challenge, which is precisely achieved through exercise, rather than attempting to eliminate all ROS with pills. It circles back powerfully to the core concept of hormesis. A little bit of stress, applied appropriately, can fundamentally make you stronger and more resilient. Too little stress, or artificially nullifying it, can leave your cells complacent and unprepared, while too much chronic stress, like uncontrolled inflammation or excessive oxidative damage, can overwhelm them. The optimal point, the sweet spot, lies precisely in the middle, the zone of mitohormesis, where transient challenge leads to profound adaptation. Mega recap and flashcards. Mitohormesis and ROS balance. Let's recap the concept of mitohormesis. We established that ROS reactive oxygen species aren't just bad guys, while high levels cause damage.
Low or transient bursts of ROS, like those produced during exercise, act as beneficial signals. This is mitohormesis, a small stress leading to adaptation.
Exercise is a prime example. The temporary ROS increase triggers the body to boost its own antioxidant defenses, like catalase, glutathione peroxidase, and mitochondrial repair biogenesis pathways, like NRF2, PGC1.
Exactly. And the crucial counterintuitive point is that taking high-dose antioxidant supplements like vitamin C and E can blunt this beneficial ROS signaling, potentially impairing exercise adaptations like improved insulin sensitivity.
So the goal isn't to eliminate all ROS, but to achieve balance. Avoid chronic high ROS, but allow the beneficial transient bursts. Exercise actually helps here long-term by reducing resting ROS and boosting internal defenses.
Right. Dietary antioxidants are fine, but megadosing pills, especially around workouts, might be counterproductive. It's about the Goldilocks principle, flashcards, reactive oxygen species, ROS, oxidative damage, mitohormesis, adaptive stress responses, antioxidant defenses, e.g. catalase, glutathione peroxidase, NRF2, stress response pathway, PGC1, biogenesis regulator, revisited, antioxidant vitamins, C and E, potential issue in high doses, insulin sensitivity can be blunted by excess antioxidants, pharmacological doses versus dietary levels, endogenous antioxidant network, the body's own system, hormesis, general principle of beneficial stress, electron leakage, source of ROS in mitochondria.
Perfect. A really important nuance for anyone interested in optimizing health and performance. Throughout our discussion today, we've mentioned various metrics and biomarkers that scientists use to assess mitochondrial function. For those who are analytically inclined and interested in actually tracking mitochondrial health, whether in a rigorous research setting or for personal health optimization, these are incredibly important tools. What are the key ways we can objectively measure the power and quality of our cellular engines? How do we know if things are improving?
Precisely. These biomarkers allow us to move beyond anecdotal observations, I feel more energetic, to quantitative, objective assessments. They help us truly understand the power and quality of our cellular engines, providing empirical data on how well they're performing and how they're changing over time, whether due to aging, disease, or interventions. Let's break down some of the key biomarkers. 1. Synthase, CS activity. CS is a specific enzyme of the Krebs cycle, the central metabolic pathway within the mitochondria. Its activity, usually measured in a muscle biopsy or a cell sample, is widely used as a proxy for mitochondrial density or total mitochondrial content. Higher CS activity generally means more mitochondria, and this often correlates well with physical fitness. In training studies, a rise in CS activity is strong evidence of mitochondrial biogenesis. It's a convenient single enzyme marker. 2. OJOS, oxidative phosphorylation capacity. This measures the fundamental ability of mitochondria to use oxygen to make ATP their energy-generating efficiency. It's commonly measured using high-resolution respirometry on tissue samples or cells. You feed them specific fuels and measure oxygen consumption under conditions simulating high energy demand, like adding ADP. You can look at individual complexes, I through OAV, or overall respiration. Higher OXPOH opacity means better energy production. It's considered a gold standard functional measure. A whole body proxy, as we've mentioned, is VO_max, since muscle mitochondrial capacity is a key limiter. 3. mtDNA copy number and deletions. Remember, mitochondria have their own DNA, mtDNA, and cells have many copies. With age, mtDNA deletions accumulate, bits of the mitochondrial genome get lost. The famous common deletion is one example that builds up in muscle and brain. High levels impair function and are a direct biomarker of mitochondrial aging, sometimes leading to visible problems like ragged red fibers in muscle. mtDNA copy number, the total number of copies per cell, is also tracked. Sometimes it goes up to compensate, but low numbers are bad. Measuring cell-free mtDNA in blood can also indicate damage or inflammation. Interventions aiming to slow aging might look to reduce deletions or maintain healthy copy numbers. We also look at other, more granular markers. Enzymatic assays for specific respiratory chain complexes like complex 1, complex 4, levels of mitochondrial proteins like COX4, cardiolipin content, which is a unique mitochondrial membrane lipid related to mass and function, and even imaging-based measures of mitochondrial network morphology. Are they nicely fused or fragmented and stressed? And for autophagy mitophagy specifically, LC32 and P62 levels give a general idea of autophagy rates, but aren't mitophagy specific. Observing PNK1 and Parkin levels or localization, like Parkin moving to mitochondria, gives a stronger clue about active mitophagy. And in research, those reporter mice like MitoQC allow direct visualization, though that's not for clinical use. Finally, on the clinical functional side, exercise performance tests like the 6-minute walk test, 6-from-WT, or full VO_max test give an integrated view of mitochondrial and cardiovascular health. And blood biomarkers like lactate threshold, the point where lactate builds up during exercise, indirectly reflect muscle mitochondrial oxidative capacity. A higher threshold is better.
So the key idea here is that mitochondrial health can be rigorously quantified in numerous sophisticated ways. ranging from deep molecular analysis in a lab to whole-body functional assessments that you might experience in a clinical or fitness setting. In any intervention, whether it's a new supplement, a structured training program, or a pharmacological drug that claims to improve mitochondrial function or energy metabolism, you would reasonably expect to see measurable and positive changes in some of these biomarkers. This could include a consistent increase in citrate synthase activity or the activity of specific respiratory complex enzymes, a tangible improvement in VO max or endurance performance, or a quantifiable reduction in markers of mitochondrial DNA damage. Without such objective, measurable evidence, claims remain highly speculative and should be regarded with a critical scientific eye. Mega recap and flashcards. Biomarkers on mitochondrial health. All right, let's recap the ways we can measure mitochondrial health. It's not just guesswork. We have objective tools.
Exactly. We covered key biomarkers like citrate synthase, CS activity as a measure of mitochondrial density or content. Then there's OX-PHOS capacity measured by respirometry, which directly assesses energy production efficiency. VO max is a whole body proxy for this. And mtDNA copy number and deletions provide insights into mitochondrial genome health and accumulated damage over time.
We also mentioned more granular markers like specific complex activities, protein levels, COX-Phe, cardiolipin, network morphology, and autophagy mitophagy markers, LC32, P62, PANK1 parkin.
And importantly, functional tests like the 6-minute walk test, 6MWT, VO max, and lactate threshold give us an integrated picture of how mitochondrial health translates to real-world physical capacity.
The main point is that claims about improving mitochondria should ideally be backed by measurable changes in these biomarkers. Okay, flashcards. Citrate synthase, CS activity, OX-PHOS, oxidative phosphorylation capacity, high-resolution respirometry, VO max, mtDNA copy number, mtDNA deletions, common deletion, ragged red fibers, pathological sign, cell-free mtDNA, COS-STOR, mitochondrial protein marker, cardiolipin, mitochondrial lipid, mitochondrial network morphology, LC32, autophagy marker, P62, autophagy marker, PNK1 and Parkin levels localization, mitophagy markers, MitoQC, mitotimer, reporter mice for research, 6-minute walk test, 6MWT, lactate threshold.
That covers the toolkit for assessing mitochondrial power and quality.
Before we wrap up, it's vital to address a crucial aspect of scientific progress itself. The nature of scientific discovery, including the often messy reality of conflicting or non-replicable data, sometimes referred to as the null replication aspect. We also need to discuss some very important safety concerns, particularly regarding NADO boosters and other supplements, especially in specific medical contexts like cancer.
Indeed. Science is not a linear, perfectly clean path. It is a dynamic, iterative process. It's perfectly acceptable and, in fact, a fundamental unhealthy part of the scientific process when some studies fail to replicate earlier positive findings. This doesn't necessarily mean the initial study was wrong, but rather that it refines and deepens our understanding, often pointing to hidden complexities or context dependencies. We've already touched on this with the mixed results observed in human trials of NDACH precursors. Many initial animal studies, often conducted using genetically modified animals or highly specific disease models, demonstrated dramatic and impressive effects that, when translated to human clinical trials, often resulted in far more subtle or even entirely null outcomes. This serves as a powerful and essential reminder to maintain a healthy skepticism and to patiently await replication by independent research groups. For example, if one small trial suggests NMN improves muscle strength, it's prudent scientific practice to look for confirmation in larger, more rigorously designed trials. If that finding doesn't repeat consistently across multiple independent studies, it might have been a false positive result, or the effect might be truly limited to a very specific subgroup of individuals, not broadly applicable.
And when it comes to the safety of NADO boosters, a particularly pertinent and highly debated topic that demands careful consideration is their potential relationship with cancer. This is a concern that should not be overlooked.
This is a complex area because NADO is fundamentally a double-edged sword. While boosting it might indeed help healthy cells repair their DNA, maintain mitochondrial function, and survive various stresses that contribute to aging, cancer cells also heavily rely on NADO. They typically exhibit a hyperactive metabolism, growing and dividing rapidly, and they have significantly increased DNA repair needs to cope with their inherent genomic instability. Therefore, there's a theoretical and increasingly experimental concern that providing an NAD precursor to someone with an undiagnosed cancer or an existing malignancy could potentially fuel the cancer's growth or enhance its ability to metastasize, meaning to spread to other parts of the body. Let me cite a recent highly impactful study by the University of Missouri which highlights this concern. Researchers used a sophisticated bioluminescent imaging probe to track NAD metabolism in mice specifically predisposed to cancer. They found that NR supplementation in mice with an aggressive form of breast cancer led to significantly higher rates of cancer metastasis to the brain compared to control mice not receiving NR. The interpretation is that the extra NAG appeared to give the cancer cells a distinct advantage in their ability to spread, possibly by enhancing their energy production, promoting their survival under stressful conditions, or increasing their stress resistance, allowing them to thrive in new environments. Now, it's crucial to provide critical context for interpreting this. This was an animal study, and it does not imply that NA causes cancer in a healthy individual who does not have cancer. However, it does unequivocally wave a significant caution flag. If an individual has a diagnosed cancer or is at a very high risk for developing cancer, taking high doses of NAGOB boosters might be risky, as it could theoretically fertilize tumor growth or progression. There are also ongoing scientific discussions about NADO's broader role in supporting DNA repair. In some cancer therapies, the goal is often to deplete cancer cells of NA, so they are unable to effectively repair chemotherapy-induced DNA damage, which is how chemotherapy kills cancer cells. This is precisely why PARP inhibitors, which function by trapping PARP enzymes and thus wasting NAD, are used in treating certain cancers. Theoretically, taking an NAD booster could, in such contexts, counteract the intended effects of these life-saving cancer therapies, making them less effective. On the other hand, the question of whether NAD boosters could actually prevent cancer by enhancing DNA repair in normal, healthy cells is a distinct, fascinating hypothesis. However, the robust clinical data to support this hypothesis is not yet available, and it's an area of ongoing research. Therefore, the safe and prudent stance, based on the current evidence, is if you are at high risk for cancer, have a current cancer diagnosis, or are undergoing any form of cancer treatment, you should exercise extreme caution with NAD boosters and absolutely consult your physician before considering their use. For the general public, NR and NMN have not shown obvious cancer signals in short-term clinical trials, but their long-term effects on cancer risk remain unknown and require continued surveillance. Similarly, with newer compounds like taurine or alpha-ketoglutarate, comprehensive human safety profiles are still being established. For taurine, the doses used in the lifespan extension mouse study were quite high. The human equivalent might be several grams per day. While we consume taurine naturally in food, especially meat and fish, and in many energy drinks, typically around 1 gram per can, and it's generally considered safe at those levels, whether chronic high-dose taurine would have unforeseen effects is currently unknown. For example, taurine is involved in blood clotting and neurotransmission, so potential impacts in those areas need to be fully elucidated. AKG is naturally found in the body and in various foods. A small human study using an AKG supplement for 4 to 6 months reported no major adverse events, but again, long-term safety data from larger populations is lacking.
This brings us back to a core principle of scientific rigor. Replication is the undeniable hallmark of robust scientific evidence. The more independent studies that consistently show a similar benefit or effect, the greater confidence we can have in that finding. For NAD boosters, some replication has certainly occurred. Multiple studies confirm that NAD levels rise with supplementation, but multiple studies also show no major metabolic change in healthy individuals. For the broad and profound effects of exercise, there are literally decades of consistent replication we know, without a doubt, that it works across diverse populations and for a vast array of health outcomes. For truly emerging compounds like taurine and AKG, we eagerly await further replication in additional animal studies and, most critically, in well-designed human clinical trials before drawing firm conclusions. This applies to any media claim of, this pill will extend your life by X%. They should be taken with very large grains of salt until robust replication is in hand.
To offer a valuable historical lesson, remember resveratrol. It was the Nanadu booster hype of about 15 years ago, widely touted for its ability to activate sirtuins and mimic the effects of caloric restriction. While it showed promise and intriguing effects in obese mice, large human trials either largely flopped or yielded inconclusive results for many of the grand claims. The scientific community, thankfully, learned from that experience. We must be rigorous, avoid overselling preliminary data, and always consider the specific context. Resveratrol might still have niche uses or be beneficial in specific disease states, but it was certainly not the panacea many hoped for. The same cautious optimism and rigorous evidence-based evaluation apply to current NAD boosters and other emerging longevity compounds. Lastly, it's fundamentally important to remember individual variability. Not everyone responds to interventions in the same way, whether it's exercise, diet, or supplementation. Some individuals might genuinely be responders to NAD precursors, perhaps those with lower baseline NAD levels due to genetics or specific lifestyle factors, or those with particular genetic profiles that make them more receptive, while others might experience no perceptible benefit at all. As we accumulate more data through advanced research, we may eventually be able to identify specific characteristics or biomarkers that predict who will most likely benefit from certain interventions. This is entirely analogous to exercise itself. Most people improve their VO2max with consistent training, but a small percentage are low responders due to genetic factors. These individuals might require a different type or intensity of stimulus to achieve adaptations. The potential for combination therapies is also exciting. Combining approaches, for instance, consistent exercise with a targeted NADO booster could potentially yield greater and more synergistic benefits than either intervention alone, precisely because exercise will effectively utilize that increased NADO in building and optimizing mitochondria. These nuances and individual differences are precisely what current cutting-edge research is actively exploring. Mega recap and flashcards. Conflicting data and safety.
Okay, a crucial reality check section. We talked about the nature of science, conflicting data, and the need for replication are normal. Not every exciting initial finding holds up.
Exactly. And we applied this to NAD plus boosters, where human trial results are mixed compared to animal studies. Then we moved to safety, specifically the potential concern around NAD plus boosters and cancer.
Right. NAD plus is a double-edged sword. While good for healthy cells, cancer cells also rely heavily on it. The Missouri mouse study showing increased metastasis with NR supplementation is a significant caution flag, though not proof of causation in humans.
Correct. The advice is extreme caution for anyone with cancer or at high risk. Definitely consult a doctor. We also noted the need for more safety data on newer compounds like high-dose taurine and AKG.
And we reinforced the importance of replication. Look for consistent findings across multiple studies. The resveratrol story serves as a good historical lesson about hype versus evidence.
And finally, individual variability. People respond differently to interventions. What works wonders for one person might do nothing for another. Flashcards. Replication crisis or challenge. Null findings. Preclinical versus clinical translation. NADO boosters in cancer, the safety concern. Metastasis. University of Missouri study linking NR to metastasis in mice. PARP inhibitors, cancer drugs that deplete NAD+. Resveratrol, historical example of hype versus evidence. Individual variability, low responders, example to exercise or supplements.
Got it. Important caveats to keep in mind when evaluating any intervention. #practical takeaways and field guide. We've covered a vast amount of complex scientific ground in this deep dive, from the intricate dance of mitophagy to the controversial question of NAD boosters. To bring it all together, let's distill this into a few clear, actionable takeaways that you, our listener, can use as a practical field guide to navigate this fascinating, sometimes confusing landscape.
First and foremost, be skeptical of simplistic miracle claims. Aging is an incredibly complex biological process governed by numerous interconnected pathways, and no single supplement, whether it's NAD boosters, taurine, or any other compound, is a magic bullet or a panacea that will reverse aging. If a product boldly claims to reverse aging or deliver extraordinary results but only presents an increase in a single isolated blood marker, such as NAD levels, without demonstrating tangible functional improvements, you should raise an eyebrow. Always ask the fundamental question. What specific functional or clinical outcome is genuinely and consistently improved by this intervention? Does increased NAD translate into objectively better endurance, measurable strength gains, improved glucose control, enhanced cognitive function, or ultimately extended longevity in robust human trials? Look for robust, replicated evidence of measurable improvements in these functional areas, not just changes in isolated biomarkers in a lab. If those are absent or the data is solely derived from animal studies, the claim is not yet definitively proven in humans. In other words, follow the golden rule. Measure what matters. An intervention should ideally improve both the NAD metabolome and the observable function. Having more NAD or even more mitochondria, the quantity, is only truly valuable if they work well, their quality, and lead to you feeling or performing objectively better in your daily life.
Secondly, a wise piece of advice. Know when to walk away from the hype. If a supplement or a specific regimen is riding a significant hype wave, generating enormous buzz and marketing claims, but the rigorous independent science isn't consistently catching up to those claims, it might be wise to hold off on investing your hard-earned money and time in it. Take NMN, for example. It became widely popular as an anti-aging pill sold extensively. However, more recently, the FDA in the U.S. raised regulatory questions about its classification, and some suppliers have made highly exaggerated claims that aren't supported by the current human evidence. If you're considering spending significant money on such supplements, stay diligently updated on the latest independent research, not just what the company selling the product asserts in their marketing materials. If, after a couple of years of scientific scrutiny and additional trials, the claimed benefits remain ambiguous or consistently unproven, you might consider investing your time and money elsewhere, perhaps in a gym membership, higher quality nutrient-dense food, or even learning a new skill. The principle here is clear. If the promise was grand, but the proof remains small, it's perfectly rational and responsible to be an early skeptic rather than an early adopter. As a general guideline, remember, extraordinary claims require extraordinary evidence.
Thirdly, and perhaps the most crucial takeaway, leverage lifestyle first, especially exercise, and do it with smart programming. The single most robust, consistently proven, and powerful way to enhance your mitochondria and significantly extend your health span is still through regular, consistent physical exercise. Ideally, this should be incorporated into a periodized training plan, meaning it's not random activity but a strategically structured approach. This means intelligently incorporating both endurance training, which stimulates mitochondrial biogenesis, and some form of interval training or resistance training, which is crucial for effective mitophagy and building muscle strength, all structured with adequate recovery periods. Over weeks and months, diligently track measurable improvements in your performance. For endurance, you can pursue a VO-max test in a laboratory setting for a precise measure of your maximal oxygen uptake. However, simpler, highly effective field tests, such as a 6-minute walk test, 6-line WT, measuring how far you can walk in 6 minutes, or how fast you can consistently run or bike a set distance, or even regularly measuring your heart rate recovery after exercise can provide excellent indications of gains in mitochondrial and cardiovascular fitness. These performance metrics have known clinical benchmarks for progress. For instance, improving your 6-WT distance by approximately 30 meters is considered a meaningful functional gain in older adults. Similarly, improving your VO max by even 5 milliliters per kilogram per minute can shift you into a lower risk category for numerous chronic diseases. Set specific, measurable targets for yourself, for example. Increase my VO max by 10% in 3 months, or walk an extra quarter mile in 6 minutes on my 6-WT. Periodization might involve a cycle of 3 weeks of progressive training load followed by 1 lighter recovery week, often called a 3.1 cycle, or consistently alternating high-intensity days with low-intensity days. This structured approach helps prevent burnout and injury while maximally stimulating mitochondrial adaptations, making your training both sustainable and highly effective.
Fourth, and equally foundational, support your mitochondria with proper nutrition and adequate rest. Beyond exotic supplements and cutting-edge compounds, do not overlook these critical basics. A balanced diet, rich in essential micronutrients like vitamins and minerals, and beneficial polyphenols, provides the necessary cofactors that mitochondria absolutely require to function optimally. For instance, iron is crucial for cytochromes in the electron transport chain, and CoQ10 is vital for electron transport itself. Adequate protein intake is also vital to support muscle mitochondrial protein synthesis, which is especially important for older adults engaging in exercise, as it aids in rebuilding and strengthening muscle.
And critically, sleep is a profound period when much of the body's repair and renewal processes occur. Genes involved in mitochondrial biogenesis and mitophagy are actually activated and regulated during the natural circadian rhythm at night. Therefore, chronic poor sleep can significantly undermine these essential mitochondrial quality control processes. Also, avoid chronic overnutrition. Practices like intermittent fasting or time-restricted eating can induce a mild state of ketosis or elevate AKG levels, which in turn might spur some UPRMT, the mitochondrial unfolded protein response, and beneficial mitophagy for cellular housekeeping and resilience. In short, the fundamental terrain of your mitochondrial health is profoundly influenced by your consistent everyday habits. Fifth, consider targeted supplementation if it's genuinely justified, and always proceed with caution. If you do choose to experiment with NAD boosters or other emerging compounds, do so knowledgeably and cautiously, grounded in the scientific evidence. For NAD precursors like NR or NMM, consider tracking something objective to assess their effect. This could involve getting a blood NAD level test before and after, though these are not yet widely standard or easily accessible outside of research settings, or rigorously tracking a specific endurance metric or subjective energy levels over the period of supplementation. If you observe no discernible difference in your energy or performance, and the broader science doesn't provide strong, consistent support for your specific context, it may not be worth continuing the supplementation. Supplementation might be more justifiable if you fall into a demographic or have a specific condition that has specifically shown benefits in robust studies. For example, NMN for individuals aged 60 and older with documented muscle metabolic issues. For taurine or AKG, they are generally considered safe in moderation. For example, taurine 1 to 2 grams per day, consistent with amounts found in energy drinks. AKG perhaps 1 gram per day, as provided by some supplements. However, megadoses should be strictly avoided pending much more comprehensive safety data from human trials. And critically, if you have any underlying medical conditions, especially cancer or severe kidney liver issues, you must consult your physician before using any of these supplements. The potential interactions and risks can be complex and serious.
And finally, sixth, keep a close eye on the research and be adaptable. The field of mitochondria and aging is incredibly dynamic and rapidly evolving. In just 5 years, we might see NADO boosters that are far more effective, perhaps discovered to be synergistic when combined with sirtuin activators, or we might uncover completely new and exciting targets. Mitochondrial peptides like humanin and MOTSC are other hot topics currently undergoing intense research. So stay curious, remain engaged with scientific developments, and be prepared to update your personal approach as the evidence base evolves and solidifies. But also remember to apply a personal N1 lens, meaning pay close attention to your own body's unique response. Some individuals might genuinely feel a significant improvement in energy on NR, while others might feel nothing but a lighter wallet. Listen carefully to your body's signals while consistently grounding your understanding in sound scientific evidence. #conclusion.
In the end, mitochondria truly lie at the nexus of cellular energy production, the complex, multifaceted process of aging and overall health. They are the essential power providers, yet they also generate signals like ROS and mtDNA leakage that can profoundly shape our cellular fate, for better or for worse.
The mitophagy, that indispensable quality control mechanism, is what maintains their efficiency. Its proper functioning appears to naturally wane with biological aging, although we have clear and compelling evidence that we can effectively nudge it back up through robust lifestyle choices like consistent exercise, or potentially with compounds like urolithin A, as they are further proven.
NADO is undeniably a linchpin metabolite that intricately ties into optimal mitochondrial function and broader cellular health. While the strategy of boosting NAD is appealing, it is clearly not a panacea. It demands a nuanced and cautious approach, acknowledging both the genuine promise observed in preclinical models and the current limitations and mixed evidence from human clinical trials.
The overarching theme that powerfully emerges from this deep dive is one of balance and adaptation. The goal is to strategically encourage ourselves to renew and adapt through beneficial hormetic stresses, such as regular exercise or smart calorie modulation, while simultaneously avoiding persistent chronic damage from inflammation or unchecked cellular decline. Interventions, whether they are supplements or potential future drugs, should ideally fit into this fundamental philosophy. They should aid and enhance the body's innate resilience mechanisms, rather than attempt to bypass or override them entirely.
As we stand at the exciting frontier of bioenergetics and aging research, the prospect of extending our health span, the period of life spent in good health and free from chronic disease, by maintaining youthful, efficient mitochondria is incredibly motivating and inspiring. However, the age-old wisdom remains as valid and powerful as ever. Move your body. This is a clear case of use it or lose it for your mitochondria. Challenge it in measured hormetic doses. Nourish it well with a balanced, nutrient-rich diet and allow it sufficient rest. If you consistently practice these fundamental habits, you are, in essence, practicing mitochondrial medicine every single day, building your resilience from the ground up.
The futuristic NADO boosters, or targeted mitophagy inducers, if and when they conclusively prove their efficacy and safety in human trials, will likely be valuable cherries on top of an already solid foundation built on those fundamental lifestyle choices. For now, hopefully, you, our listener, have gained a much clearer and more comprehensive picture of how mitochondria and mitophagy intricately factor into the aging process. And critically, you are now better equipped to interpret the often exaggerated hype and the genuine hope surrounding compounds like NADO with a more informed and critical eye.
Whether you are literally preparing for a challenging mountain trek or simply navigating the metaphorical mountains and valleys of life's journey, diligently tending to your mitochondrial health will fundamentally ensure that you have the enduring power and resilience to not just survive, but truly thrive.
The deep dive into our cellular powerhouses truly reminds us what profound power lies within us and how understanding it can profoundly shape our journey. Keep exploring, keep questioning, and keep driving your own understanding.